The sliding filament theory. Ever wonder how your muscles move and why they can do it? Here's a skeletal muscle. These are muscles that we consciously use, like biceps brachii or pectoralis major. If we zoom into the muscle, we will be able to see how, step by step, your muscles are able to contract and move. This long and thin fiber is called a myofibril. Myofibrils hold multiple contractile units called sarcomeres that run lengthwise adjacent to these myofibrils. Thousands of myofibrils are squished together and form an individual muscle fiber cell. Thousands of muscle cells are also squished together to form a single muscle fascicle or bundle. A skeletal muscle is composed of multiple muscle bundles. Muscles will contract or relax when they receive an electrical signal from the brain or nervous system. Each muscle fiber depends on the axon, which is a neurotransmitter, to pass a signal, causing a contraction. The place where the axon terminal and muscle fiber connect is called the neuromuscular junction. Okay, remember the sarcomeres? The sarcomeres are composed of paralleled, alternated, thick and thin myofilaments, which are the ones who make every single muscle movement possible. A muscle contracting is all about these thin filaments called the actin to slide past the thick filaments called the myosin. This motion shortens the distance in between each Z-line, which is the borderline of each sarcomere. As the two Z-lines of the sarcomere come closer, each contractile unit does what it is supposed to do, contract. Each myosin has many tiny myosin heads which desperately want to bind to the actin strand, but this process needs many components to take place. Each muscle fiber cell is surrounded by a sarcoplasmic reticulum, which is the cell's storage and transport system. When the body sends an action potential through the axon, it synapses with the muscle fiber cell. The receptors in that muscle cell are sodium channels. So when the motor neuron releases acetylcholine into the synapse, the sodium channels are opened up and create a rush of sodium into the cell through the sodium channels. That same action potential travels through the muscle cell's membrane, the sarcolemma, which has lots of tubes that go deep inside the cell called T-tubules. When the action potential travels through these tubes, it eventually triggers voltage-sensitive proteins that are linked to calcium channels also in the cell's sarcoplasmic reticulum. This will open up the channels and let calcium ions flood the inside of the cells in a process called depolarization. Now, back with the desperate myosin heads which want to bind with the actin, they face a big problem. The binding sites of the actin are blocked up by two proteins called troponin and tropomyosin. The sliding of the myofilaments require many conditions. First of all, the actin requires calcium ions to bind with the troponin, causing these two proteins to be displaced, enabling the myosin heads to bind to the actin binding sites. This is where the depolarization and presence of calcium ions become vital to the sliding of the filaments. But the myosin heads can't bind to the actin binding sites just yet because they require ATP, which is the energy currency of the whole human body. As an ATP molecule binds with the myosin heads, it is hydrolyzed into ADP and phosphorus, giving the myosin head enough energy to bind to the actin and perform a stroke movement in which the actin filament will slide and therefore contract the muscle cell. As calcium enters the cell, it binds to the troponin, which causes a displacement in both of these proteins, revealing the binding sites of the actin. So as all of this happens at once, the myosin heads are able to bind to the actin, and with the help of a little energy from the passing ATP molecules, the millions of myosin heads perform the stroke movement and slide to the actin parallel to the myosin. This is how the myofilaments complete a successful contraction in the muscle cells, by reducing the distance in between each Z-line of each sarcomere.